All right. Hey, everybody. Welcome to our, I don't know if this will be the inaugural, but we're going to try to make some quick 3 to 5 minute videos on how to analyze your data in more depth using Spectronaut. And so I thought I would start out with a good example. This I just did yesterday, so I thought I'd do it fresh in my mind. I don't actually know when I'm going to publish this because I think we're going to wait for the paper to be published. But this was with a group that we've been working with, and we are in the middle of publishing the paper, and one of the reviewers is asking us for basically some evidence that their controls were not contaminated with some of their preparation proteins. So some of the proteins we're looking at today are called LAMP proteins, that's lysosomal associated membrane proteins. And we kind of just want to figure out if they're only in our experimental and not in our control. So what you're looking at now are two of these proteins we found in our data set. There's one, this lysosomal associated membrane glycoprotein 2, and there's another one underneath it, which is, let's see what that one is, lysosomal associated membrane glycoprotein 1. Okay, so we kind of want to figure out if they're in our controls or not. What we're looking at now is our Simplify results. So these are the results we take from Spectronaut, put them in the Simplify. They make them easier for biologists to look at. They kind of cut out all of the mass spec stuff. But for this, we kind of want to look in depth in the mass spec data and see what we can find. And so for this example, you can see this LAMP protein is only found in our experimental and is not in the control. You can see there's question marks here, and this is just because Spectronaut puts in filtered instead of NA. And the other protein is only found in the experimental, and some are found here, but we're going to look at this in depth to try to figure out what's going on. Okay, so we want to figure out if these are only in experimental. They seem to be, but let's look at the mass spec data on Spectronaut. Okay, so let's do that, and I am going to switch my, let's see, where is, I'm going to switch, let's see here, main display, window capture, let's do this one above. Okay, so now I am switching to the Spectronaut results, and so let's look at that. I have those open here. First thing we want to do is try to look for those proteins. So we go to Analysis, and Grid View is the easiest way to do that. Let's type in the first session number, which is P13473, and there it is right there. And you can see that these are not organized correctly, how our experiments are all kind of spread apart, and we want them together. So the first thing we want to do is go over to Tree View, and we go Organize, and let's see, where is it? Order runs by condition replicate. So this will put all the conditions together, this condition with the control and that one. And so now we're going to go back to our grid view and type this in again, and they should all be ordered, and they are. Okay, you can see there's no evidence for them in any of our other experiments, but what we want to do is see how many peptides we see here. So we're going to right click on here and put column chooser and pick number of peptides. We'll put that here, and we'll close it. And you can see, unfortunately, this is only identified with one peptide, so not the best. So let's see if this is if this is a really high quality result, or it's kind of sketchy. So let's find out. So this is going to kind of show you the coverage. So we have one peptide here, GILTVDELLA. If we click on here, one of these numbers under our experimental condition, we can see it kind of looks pretty good. We have a nice transition. Unfortunately, we can't, I don't think you can click on this to go to the grid view. At least I don't think we can. So we're going to have to memorize this peptide, or I'm going to write it down. So it's GILTVDELLAIR. We don't need all of it, but that will help us. We go to tree view, and we want to pull out this identification. So we'll do the drop down and say sequence, which should be peptide sequence here, and then we type in the sequence GILTVD. TVD, there it is. Let's see. There it is here. Why it's not showing up there is GV. Was that our sequence? It should be in all of them. And they're not. And let's figure out why. GILTV. I spelled it wrong. Okay. So no, I didn't. GILTV. And why they're not showing up in this one, LLUME. It's interesting. It is in here. GILTVD. TILTVD should be here, but why is it not in LLUME3 when it was before? Let's go look. Okay, and there it is. It is there. Did I just mess this up by clicking on it? I probably did. Let's see. GILTV. GILTV. And why is that not? There we go. GIL. I don't know why I did that before, but now it's all in each one of our runs, and we can see it here. There it is in that one. And there it is in that one. And there it is in that one. It looks pretty solid. Let's just look at it in this view. See, I think I messed it up. Let's see, why is that different? There we go. Looks a little bit worse in this one, but pretty solid in these. So what we're going to do is we're going to view it with, let's see, we want to do across runs, MS2, let's do XIC, let's do cross run. RT accuracy, this one looks pretty good. So you see how these boxes are kind of lined up here. They're green. They're all kind of together, where they're kind of all over the place in the other one because there's really no evidence for it. We also want to look at, I believe it's the XIC graph. Doesn't look so great on the MS1 intensity, but we see very good signal for the MS2 intensities. They all kind of line up. There's no real evidence here. I'm pretty happy with this result. So it looks good. Looks good. Looks good. Everything looks good. I don't know why it didn't turn out the last time, but I'm pretty happy with this. Okay, so we have pretty good evidence that this peptide, even though it's only one peptide, is only in our LLUME samples. So pretty good. Looks kind of noisy here, but I think it's decent. And we can even go to the MS2 and MS1, and you can see there's not a lot of MS1 here, but the MS2 looks pretty solid. Okay, so let's look at our second protein, and that is P11279. That one shows up. You can see how there's solid evidence for it in the LLUME samples, but you see some does show up on the these other group of samples here. If we turn off the log scale, you can kind of see more how intense they are in the LLUME and not very intense in these samples. So in this protein, we have 3 peptides, so it gives us a little bit more to work with. Let's look at them by clicking on here. And you can see kind of the peptides. This one doesn't look very good. These 2 look pretty solid. We could do, let's look at the AL, Q, A, T, V, G, and SYK. Unfortunately, you can't click on it, but I'm not going to click on it because I kind of messed up I think it messed something up last time. So we're going to type that in here, ALQ, ATV, and will that be enough? And it is ATV. And you can see it's coming up in the ones we think they are, ATV, G, N. Okay, boom. And you can see that here. Let's see, we click on this. Looks pretty solid. This one looks really good. So we got a lot of MS1 signal. We got a lot of MS2 signals. We will look at the cross runs RT accuracy. That looks pretty good. See how they're all lined up in the LL, those samples we think they are. We do have something that looks like it could be in in this sample, number 3, maybe in sample 5, but you see how the aligned retention time is way off, so that's probably not a good result. So let's look at the XIC graph, and that will give us a little more information here. So you can see how they're all lined up, the MS1 intensities, MS2 intensities, and our LL UME samples. You can see how they're really all over the place and not looking very good. And the other samples, even though it said it was actually in control 3 and ASO2 3. And so here's control 3, and we don't really see much signal here. So that must be really borderline. And we can look at that here, and that does not look good at all. And you can see the difference. Like, that's really like just picking stuff out of the noise. It has a Q value just below the cutoff, 5 times 10 to the negative 3. But you can see it's really not a good signal. So I'm really happy these are in LUME and not in the other ones. We don't see any evidence for it. So one of the things we can do is, one of the reasons, if you go back and look at the Spectronaut, the Simplifi results, this protein was not shown to be differentially expressed. But in this sample, it really looks like it should be, right? Because it has strong signal in LUME samples, but kind of sporadic in the other ones. So why is this not differentially expressed or reported as significantly differentially expressed? If you go back and look at the reports, actually we'll go to candidates and we'll type this in, P11279, and I just wanted to show you this example. You could see it's differentially expressed, or it's below the cutoff value, and the AAOS versus the LUME, but it is not in the control samples versus the control samples. So we really would like to see it differentially expressed in the control samples, and it doesn't look like it is. It's not matching our cutoffs. So why is that? If we go back, my suspicion, I've already done this, so I know. My suspicion is that the t-tests, they have trouble with missing values, and there's kind of a sporadic signal here, which we know is not true. It's kind of background. So my suspicion is that because of all the missing values and the little signals here, it's not showing as differentially expressed using the t-test that Spectronaut does, even though your eyeballs clearly look at it and you're like, duh, it really is differentially expressed. All right, so how can we fix that? So we can give it a little boost by turning on imputation, and that sometimes helps with t-tests. So what we do is we go back to this experiment, we go settings, identifications, or quantitation. Instead of Q value, we do Q value sparse, and that turns on global imputation. And remember, you can always go back and pause this, and because I'm going a little fast. And then you go confirm, and you just give it a minute. And it doesn't actually take very long. So let's give it a minute and see. Almost done. And you can do this with the viewer, by the way, so don't worry about it. Okay, so done. It's done. Okay, so let's go back and type in P11279. And there we go. And you can see imputation is turned on, so we all have a signal here. Basically, Spectronaut does a very fancy form of imputation. That kind of gives everything a low signal. I think it does like a sampling within the 3 standard deviations away, a random sampling, I think. This is all in the manual. I should read it again. Clearly, by my eyeballs, I say it's differentially expressed. If we looked at a box plot, you could see the whiskers were far apart. So the t-test is going to be significant. But let's go back and see. Go to post-analysis, candidates. And let's type in that signal again, that protein again, P11279. Bam. And there it is. First, let's see, where's the control? There it is. I bet it's right there. Yep. So LLUME versus control. Clearly differentially expressed. It'll make things look better if you turn off log scale. Okay, so clearly differentially expressed in the LUME, not in the other samples. It really isn't in the other samples, even though the imputation now makes it look like it is, but remember it's imputed. And I wish they would, in the next version, go back and recolor these, or like make a little arrow next to them that says imputed, because that would, sometimes imputation really confuses biologists. And it confuses me sometimes. 379. We'll type in our first protein, P13473. Bam. Okay. There's that first one we did. Sometimes it takes a second for it to 73. And you can see they're kind of, you see signals here where you haven't. So you can see how when you turn on the log scale and not use the log scale, it makes it look is really different. I like turning off the log scale. And remember, this is just that one peptide. We can go back and look at it again. Okay, let's go back and to analysis, tree view, peptide sequence. Where is peptide sequence? And we'll type in that first peptide again. Don't remember what it was. Let's go back to, let's see, let's go back to our tree view, which is here, our grid view. And 3473 is GILTVD. Let's do that again. GILT, was that it? GIL, you can see how well my memory is working today. GILTVD, TVD, C. And there we are. They only show up in our LLUME samples. I must have screwed it up the first time somehow. Okay, and we want to look at this. Looks good. We could do the MS2 and MS1 on the bottom. Oops, where is it? MS1 run specific, MS1. You can see we don't have very good evidence for the MS1, but the MS2 is strong. And so, and it all lines up. So I'm pretty confident. Okay, so anyway, this is how to go through some of your Spectronaut data relatively quickly. I thought it would be quicker, but sorry about that. But I'm pretty happy with this result. We've shown that the one protein was only identified with one peptide. So this shows you that sometimes it's good to filter those away, sometimes it's not. In this case, it was good to keep them because we actually showed some good mass spec evidence that this protein was not in our controls and not in our other conditions, which is good evidence that the experiment was done correctly. Okay, anyway, thanks again. Sorry this took a little long, but I'm going to try to have shorter videos. I'm hopefully going to publish this online once the paper gets accepted. Hopefully it'll get accepted. I think this is pretty good evidence that these proteins are only in the LUME samples and not in the control. And if you don't believe me, you can download the data and follow my videos and see if you could come up with something different. But that's my analysis. Okay, all right, thanks everybody. Sorry this took a little long. We will be back hopefully soon with some more bite-sized videos about Spectronaut and analyzing your data. Okay, thanks everyone. See you next time.